out, and it looks like everybody has their mics muted, which I certainly appreciate. If you are not actively speaking, one of our presenters, I do ask that you mute your mics just because the interference from other people's computers can really affect the sound. If you have any questions at all during anybody's presentation, feel free to type those questions into the chat box. I will be looking at those throughout the entire time, and after each speaker is done kind of presenting to you, I will be addressing those questions to them for everybody to hear. So that's just basic housekeeping. You're welcome to turn your camera on if you would like, but no pressure to. I know that this is pretty much the standard kind of look in meetings, but don't feel like you don't. If you want to show your face, please feel free. Okay, so my name is Miranda. I want to introduce myself first. I am a newer board member with the Horned Lizard Conservation Society starting this year. I serve right now as the director at large, and I've just been loving being a part of this wonderful group. So I'm going to be moderating for today. This is our first state of horned lizard conservation, and we're focusing on the state of Oklahoma, which is where I live and where I did my master's research on horned lizards recently. Okay, so I know that a lot of you are probably already members, but I just want to throw that out there, that if you're not already a member of the Horned Lizard Conservation Society, you're free to go to our website, hornedlizards.org, and join membership. It's very cheap, and it helps to conserve horned lizards everywhere. So just throwing that out there for you if you haven't already done so. Okay, so in Oklahoma, we have a quite active Texas horned lizard conservation program that works together with a lot of different people. So we have people from the University of Oklahoma here in Norman in central Oklahoma, as well as a large project that's been going on for a number of years at Tinker Air Force Base in central Oklahoma as well. And in collaboration with the Oklahoma City Zoo, we are also doing a head starting program. And then we're also starting to do some surveys out in western part of the state to find out where horned lizards are, to see what we can find. And that just started last summer. So I have people here that are going to talk about all three aspects of that and how we're all kind of connected. Yeah, so that's kind of what to expect. What we're going to do here is we're going to start with one presentation at a time. And give them an opportunity to show you some slides or some pictures or just talk to you about what's going on. And then, yeah, as I said, put those questions in the chat box for you. So first, I would like to go ahead and start with Raymond Moody. So Mr. Moody serves as a natural resource scientist at Tinker Air Force Base in Midwest City, where you can feel free to start sharing your screen at any time. So he serves as natural resource scientist at Tinker Air Force Base. He earned his Bachelor's of Science degree from the University of Oklahoma in zoology, and then earned his Master of Science at OU in environmental science and fisheries. He has taught in the past as an adjunct professor teaching zoology, environmental science, and geology. He has over 30 years of experience working in the Natural Resource Department at the Air Force Base. And in that time, he has explored, searched, and studied the prairies, woodlands, and streams of Tinker and the surrounding areas, documenting new species for the Air Force Base. Rhea has pioneered research on the Texas horned lizard in Oklahoma for the last 18 years, discovering and publishing previously unknown biology and life history of this state-sensitive species. He is published in a number of professional journals, such as Journal of Herpetology, Herpetologica, et cetera, no need to name. And he recently served as the Oklahoma on the Oklahoma Department of Wildlife Conservation's Technical Committee for potential listing of the Texas horned lizard. And he is also our current HLCS regional contact for the state of Oklahoma. So all the people in Oklahoma have any questions, concerns, or need to reach out to anyone, either myself or Ray are your points of contact here. So that is Ray. On a personal level, I've gotten to work with him since 2014, so a number of years, so we've gotten to know each other well, and I'm excited to have him talk to you a little bit about this. So I'm going to go ahead and mute now and let Ray take over.
All right. Well, thank you for the introduction, Miranda. I hope everybody can hear me. Are we good on that? And you can see my screen? Fantastic. Well, it's really an honor to be able to talk to everybody tonight. And I thank the Horned Lizard Conservation Society for allowing me to do this. So I hope the length of our study that shows up here at 18 years doesn't date me too bad, but we have had a chance to have a lengthy study on the horned lizard, which is somewhat unusual, I think, to have this long of a running study. I never thought it would go this long. So one of the slides I always start with is this one. And this thing has kind of evolved over the years. Different organizations kind of swing around and change occasionally, but the bring home point I'd like to talk about this slide is the partnerships and the way partnerships create synergy. If you look at the definition of a synergy, it's basically the interaction, cooperation of two or more organizations that create a greater combined effect than the sum of their separate effects. So if you look at some examples here, just the longevity and the organizations that have been involved, all the way from the Corps of Engineers that we have Pat Gerton on the line. I saw him up. Been helping us administer the contract for over 17 years on this, all the way to volunteer organizations, private entities, and universities, which have really been key for our research. If you look at the funding that kind of comes along with these partners, it's varied and it's wide ranging. So definitely the take home message here is, partnerships allow us to do a lot more that we can do together than we could have ever done on our own. So I thank all those who have helped us out over the years, and they really have made our project successful and the longevity of them. Here's an example right here out on our prairie site of we have represented in this shot just last week 2 federal agencies, a private organization, and of course Oklahoma City Zoo and OU researchers. It's a great example of our partnering. I wanted to go over just give you a little bit of history of our study. I won't go into much detail here, but you can see we started out in 2003 really just trying to find out all about the biology of horned lizards. We knew very little. So our first thing was to start tracking these guys around and find out about them, find out what their demographics were, what their habitat requirements were. Since then, we've gone through various universities we've partnered with. And one key thing you'll notice we had throughout these phases of the study is we've wanted to track and monitor our population. We wanted to make sure that the population was doing well and was stable. Along the ways, we took a lot of sidetracks and learned a lot of different things about the horned lizard biology and its population. One of the things we found later in the mid-teens was recruitment was very important. And so in order to understand recruitment, you really have to track subadults. And we couldn't do that with the radio telemetry we started with. So we started looking at different technology to track horned lizards, all the age classes from hatchlings, juveniles, and adults. More recently, as we've been able to do that kind of work, we found that we needed to learn more about translocation and how effective that could even be. There's a lot of research that says it's not effective, but it would be a great tool to be able to utilize head starting and translocation. And so a lot of our objectives have been focusing on that in the last couple of years, and I'll go over that a little more. And also with the emergence of wildlife disease, the health of our population and individual lizards has also become an important thing to look at and focus. So this screenshot gives you a little idea of what we've done so far. We've captured over 1,089 individual horned lizards on about a 40-acre site. Each one of those dots you see on the screen, red dots, represent a Texas horned lizard, either initial, a capture, or a recapture. Each of those dots also have a wealth of information about everything from the vegetation type it was on to what behaviors it was doing. All these data are put into an animal database that we have on Tinker. So that represents, those dots represent over 40,000 captures and recaps. So what is the status of the Texas horned lizard? Here in Oklahoma, it's a state sensitive species. The Oklahoma Department of Wildlife recently did a, well, it's actually been a few years, but they've done a Oklahoma conservation strategy and that identified species of greatest conservation need. The Texas horned lizard ranked high in that, and so that is Oklahoma's state ranking as a state sensitive species. I know in Texas, the horned lizard is state ranked as threatened. Here in Oklahoma, we did recently, I was on Oklahoma Herpetological Committee, and we did a rank assessment of the horned lizard. It had previously been ranked as a S2 species. As we went through the rank calculations, we came back and found that even with the declines, particularly the eastern parts of the state, we're able to track based on observations and everything from museum findings to every kind of sighting of a horned lizard we could come up with by county. We're able to see how the range had slipped farther to the west. So the S2 ranking for Oklahoma, I feel, is a good number. So that the definition of that is it's a species at risk of extirpation in Oklahoma due to restricted range, few populations or occurrences, or steep declines and severe threats and other factors. So the biggest factor with the horned lizard is it being an animal specialist, particularly with diet. As most of you probably know, the horned lizards primarily feed on ants. That makes up over 90% of their diet. So that's one thing they have going against. The other thing is they really require a specific habitat. They need a mosaic of vegetation types. One of the things we noticed kind of early on is they needed bare ground as well. That was really important to that vegetational structure. So I'll talk a little bit about this methodology and the way we tracked horned lizard, give you an idea of how we've accumulated all this data. First, we started tracking adults with radio telemetry. Later, we moved on and started looking at how we could track hatchlings and juveniles. We used harmonic radar. And more recently, working with OU, we're looking at cellular tracking technology using solar tags. So here's the 3 age classes of Texas horned lizards: a hatchling, a juvenile, and adult. And those are all outfitted with one radio telemetry, and the 2 smaller juvenile subadults are outfitted with harmonic radar diodes. And we'll get into a little bit more about that. Just some of the radio telemetry type equipment, some of the technology we use. For adults, we actually microchip our horned lizards using pit tags. And again, we GPS our point locations using a Trimble unit that has our attributes for each data point. Some of the other things we do in the lab when we catch an adult lizard, we weigh them, we measure them, do all the morphological measurements. Those are entered into our database as well. So here we are out in the field doing radio telemetry. It's tedious work, but radio telemetry is very effective, and it's got good range, and it was very useful, and being able to find adult lizards were fairly easy. So when we go to the harmonic radar, just give you an idea of what that is. Harmonic radar uses a electronic diode and a handheld radar unit, and it's actually used in finding avalanche victims. RICO is the name of the Swedish company that developed the technology. And we kind of partnered with them and helped develop our tags for our lizards. As you can see early on, we kind of designed our own diodes. Here's some shots of them. They look a little primitive to me now compared to what we have. They were pretty cheap, but they were hard to make. I had to learn how to micro solder, and that was not easy. I didn't have a lot of time to spend making a lot of those, so we're able to work with the Swedish company to actually, they had some diodes that they have a plastic strip, and they were able to modify those slightly to make those a little more durable and put some paint or camouflage on them, and they actually worked great for us. And so we were able to start just getting those directly supplied from that company. And there's a shot of us out in the field using the harmonic radar. The range on harmonic radar is only 2 to 6 meters. So you have to be a lot closer than with radio telemetry. So you have to be in the field more, which takes a little more labor, but luckily, at least the hatchlings and subadults don't move as much as adults do. So if you're consistent, it can be a practical way to track subadults. One thing we noticed as we started tracking these hatchlings that they're much more fragile than adult lizards. We actually, after losing a few lizards, we actually brought our processing into the field and processed lizards on site, taking the same kind of measurements and recording the same data, but we actually put these diodes on right in the field, and we use super glue, gel super glue to put those on. We also marked horde lizards. We did everything from painting dotted codes on their bellies to eventually we came around and just used Sharpies to mark a number on their bellies, which seemed to be the most efficient. So how are we tracking now? So we are looking at cellular tracking technology using these solar tags. So they have the advantage of being lighter weight, which is great. And the problem is if they don't have light, they don't have power. And so if a lizard burrows underground or you can't do any night tracking, so there's a disadvantage, but a lot of those are being outweighed with just not the length of how long these will go. We haven't really got a good handle on it, but it's definitely longer than the longest radio transmitters we have, which were about 9 weeks. So these solar tags can go indefinitely. So we're kind of testing those to see how well those work. There's one of our little guys out there with if you look closely, you can even see an ant on that solar tag. So Brenda said I might want to share some of our findings, and we do like to do that as we're partners with a lot of organizations across the state. We have the opportunities to do that. So I thought I'd highlight the most interesting things we've come across, and then some of the more, we'll go back to the 2 basic things, habitat and their diet is kind of focus on the things we found there. So again, here's their habitat. Bare ground, I used to have the text for bare ground real small in this slide, but I made it bigger this time because the importance it is. But our lizards use a mosaic, we'll say, of vegetation. They need wood, they need shade. So things like forbs and bushes and small trees are useful for them. They need prairie grass to escape from predators. So they need a mix of all this together with the bare ground. Bare ground is important for a couple of reasons, for movement and also for thermal regulation. These guys are cold-blooded, so they need to get out and get the sun. And so that is the structure of habitat is probably the most important thing we've found for their habitat. So what about their diet? We all know they're myrmecophagic, meaning they primarily eat ants. We did a study of ants, and we found we had 17 different types, genera of ants on base. Surprisingly, we didn't have any harvester ants, which most of the literature says that's what horned lizards feed on. We found that there were 12 different species of ants in the scat of the horned lizard we had in Reserve 3. Those 12 species of ants, there were 3 that stood out. The acrobat ant, wood ant, and big-headed ant were the predominant species in their scat. Those 3 ants happened to be larger ants, some of the larger ants we had as well. So likely adaptation on the size of their prey was in play there. Nesting sites. As we tracked lizards to their nesting sites, a couple things here. One thing we notice is females often traveled pretty good distance before they start digging test holes and digging nests. We found they dug about a 5 to 7 centimeter depth and had a clutch size of around 17 eggs. Hibernation, as we tracked our lizards into hibernation, surprisingly, they didn't burrow as deep as we might have thought. The other thing we found, primarily them hibernating on southwest slope aspects. Also, length of hibernation was about 5 to 7 months. The thing I could set my clock by is by them emerging. Pretty much the first week of April, our horned lizards are out. Within a week of that, seemed like no matter what, over these last 18 years, that's when they come out. When they go into hibernation, that was quite different. It's kind of a range of timing. They would slow up in the fall as temperatures cool, but we had lizards that stay out as long as December before they would burrow in at all. Some of them would be buried up in October and would not move again. So that's an interesting thing. We're not sure why that is. Sexual dimorphism. So here's a male and a female. The females are much larger than the males. The other thing with size, we see here as you go north, we see the horned lizards are much smaller than they are as far as like down into Texas. Typically, our lizards 2.5 to 3.5 inches, 3.5 on our larger adults is about their typical size, and they get much larger than that. And so that's opposite of the mammal rule of Bergman's law. For reptiles, it's opposite of that, and so they hold true with that. So let's talk a little bit about the hatchlings. As we started tracking hatchlings, we found some really cool things. As these guys emerge from their nests and come out, as we track these guys with harmonic radar, one thing we found is we would go out, we'd find a hatchling, would come back the next day to the same location, would expect, these guys' home range is only about maybe 10, 15 meters in diameter, and would come back out and they wouldn't be there. Come out the next day, they'd show back up. Finally, we noticed they were scurrying into the desiccation cracks in the clay. We have heavy clay soils, and so we can get cracks in the summertime that go down 24 inches in depth. So we actually observed these guys going down into these cracks, one, to escape predators, and secondly, it's a lot cooler down at the bottom of these cracks. There's about a 3 to 5 degree temperature difference from the surface to the bottom of the cracks. So that was an interesting thing we found. The other thing is we tracked the hatchlings to what we thought would be hibernation. We found these guys weren't burrowing into the ground at all. They literally lay on top of the ground as hatchlings over the wintertime. Whether they actually hibernate, I'm not sure. We actually had lizards make movements. Not sure if they're blowing around. They did only move a foot or two, but that's something left that we can look at. The other unusual thing, if you can see the yellow lines, these guys are very cryptic, just like the adults. You can actually see in the lower left-hand corner, there's actually lizards stacked on top of each other. One clutch of lizards that we had in one area, I recorded 3 to 4 lizards stacked on top of each other. And I don't even know what that's about. So the other cool thing is snake predation. So in the past, we've tracked horned lizards from all over our sites. One thing that occasionally happens is as you're tracking, a lizard will seemingly be moving much faster than a normal lizard. Pretty much anytime that happens, you probably have some kind of predator. These are a couple snakes and x-rays we took of speckled king snakes that have munched on our lizards. You can actually see in the x-rays, you can see a cellular tracking tag and also a pit tag. The last speckled king snake actually ate at least 2 lizards because those were 2 separate tracking devices that we had on that lizard. Another event we had was one that was a copperhead. That one was fun to run into at the end of our tracking session. So back to the monitoring thing. What are we seeing over the years? This is our population trend line since 2003. You can see the green line as it dips in around 2009, 10, all the way into 13. Around that time, we know there was some habitat loss. And so we feel like that's probably the reason we had a decline there. But the interesting thing, the hopeful thing, is you can see that quick rebound in 2015 and 16. And so our populations right now seem to be pretty steady, actually even a little higher than what they were back in 2003 and 4. If you look also on this metric, it kind of shows the yellow squares, which we didn't have survival data for every year, but that survival data follows the population line quite well. So this is a big thing for us, and to be able to track this populations and make sure that we're not seeing big fluctuations or problems. It gives us an indicator of our ecosystem health, actually. So it's a good tool for us to use as managers across, and even hopefully it could be used across the state. So with that, what does the future hold? Well, hopefully a lot of these little guys. We just awarded a new 3-year cooperative agreement with OU. So we're going to continue work with them, and we'll just see where that leads us. Hopefully our lizards are in good hands, and I think they are. That's it.
Excellent. Thank you very much, Raymond, for sharing that with us. You can feel free to turn off your screen. Great. So that was excellent. Like I said, feel free if you have any questions that we can address at the end. Feel free to add those in the chat box. If you look in the chat box right now, you will see that a couple of links have been posted. One is for the Wildlife Department here in Oklahoma, where you can report any Texas horned lizard sightings. So if you see a Texas horned lizard in the state of Oklahoma, please report it with as much data and take a picture as well, if you can. That helps to have us get a better idea of what's going on big picture in our state. And there's also some links for the 2 different kinds of equipment that Ray mentioned in his talk, both the Reco and the newer tracking technology with Cell Track Tech. So those links are in there if you are a researcher looking for some sort of new method for tracking. Okay, so I'm going to go ahead and introduce Sam. He's going to be our next presenter. Sam is a former coworker, lab mate of mine at the University of Oklahoma when I was studying horned lizards with Ray as well. He is currently a Ph.D. candidate in Cameron Seiler's lab at OU. He has broad research interest in applied herpetological conservation. His current work focuses on Head Start and reintroduction programs and ways to improve reintroduction success for imperiled species. He is particularly interested in better understanding how gut bacteria in individuals pre and post-release affect reintroduction outcomes. Sam first began working with Texas horned lizards on Tinker in 2018 and has been running the day-to-day operations of the Lizard Lab, it's what we call our Head Starting program, at the Oklahoma City Zoo and Botanical Garden since its inception in 2019. Outside of horned lizards, Sam has published research to better understand and preserve a variety of herpetological taxa, including geckonoids in the Philippines and Fijian iguanas. Also, he is one of our HLCS grant recipients for 2021 as well. So I will go ahead and let him take over at this point and talk to you a little bit about his work.
Thank you so much, Miranda. Let me just share my screen here and make sure that's working okay. Okay, is everybody able to see that? We good? Wonderful. Well, as Miranda said, I'll be talking about the Head Start and Translocation program that's been running in collaboration with a number of institutions across the state of Oklahoma. And we'll start off with why we basically began a Head Start program in the first place. And so, as Ray touched on with a few of his graphics in his slide, Texas horned lizard survival rates have been well monitored on Tinker for a number of years. And what was a particularly interesting finding to me in the study from 2014 was looking at survival rates across age groups. And so we can see that adults generally survived on Tinker WR3, this specific population that we've been working with. Adults will live, will have about a 50% survival rate year to year, juveniles only 25%, and hatchlings only 33%. So we can see extremely limited survival rates, particularly in the younger age cohorts. And what that same Wolf et al. Study from 2014 found was that hatchling survival was the top ranked parameter for population expansion on Tinker. And the authors even said explicitly, essentially, that a Head Start program might be highly beneficial for expanding a population in a species with low adult survival and high fecundity like Texas horned lizards. And so with that in mind, when the opportunity arose in 2019 for an NSF project or a National Science Foundation project to be funded, myself and other researchers at the Sam Noble Museum got in touch with folks over at the Oklahoma City Zoo and with Ray and other personnel at Tinker Air Force Base to develop what we call the Lizard Lab, which is essentially a Texas horned lizard Head Start and reintroduction program. And so what we've been doing since 2019 is a pretty simple blueprint to try to boost the population on Tinker. As Ray said, you know, with that dip in 2014 with an ephemeral population, just a really bad season can be a tremendous decline in the population in general. So we want to make sure that we have the capacity to ensure horned lizards continue to persist in our area into the future. So the basic layout of the Head Start program, what we've been doing for a couple of years now, is collecting wild eggs or collecting hatchlings immediately from Tinker upon their emergence, raising them in human care in the lizard lab at the Oklahoma City Zoo, and reintroducing those individuals back to Tinker Air Force Base when they're at more robust sizes to hopefully increase their survivorship and reproductive capacity in the wild and Tinker. And while doing that, one thing that's been of particular interest to me is monitoring gut bacteria in lizards pre and post-release, so we can get a sense of how certain bacteria in the gut may be impacting the lizards while they're in human care, and how that affects their ability to acclimate to different conditions when they're moved to Tinker. And so, as Ray touched on, none of this would be possible without a tremendous effort from a litany of organizations. So we've been funded by quite a few folks, starting with the National Science Foundation, and then the Oklahoma City Zoo, Tinker Air Force Base, the University of Oklahoma, and the Horned Lizard Conservation Society has been a tremendous help as well. And we have a great array of partnerships from the Silo Lab at OU, the Zoo, Tinker, and more recently the Oklahoma Department of Wildlife Conservation as well, which has been a tremendous help. So none of this would be possible without a wide array of organizations and personnel all working together to kind of bring this Head Start project to fruition. So I'll break down now step by step, essentially what's gone into the development and implementation of this Head Start program, and then talk about some future plans. But it all starts with finding and collecting eggs on Tinker. And so Miranda was an absolute hero at developing this initial protocol in 2019. And one of the wonderful things about working with such a well-studied population is that we already had the tracking capacity in place to find gravid females. And so I would be able to go out with Miranda and other trackers on Tinker and notice when gravid females had dropped significant amounts of weights overnight, and then we would be able to go out and find nests and excavate those. And so we could collect a number of eggs, ultimately bring them back to the lizard lab where we could incubate them in an artificial incubator at 83 degrees and around 80% humidity for 60 days in reliable incubating media. And ultimately, we were able to hatch out quite a few lizards from this protocol over the past couple of years. And this is a general overview of some of the husbandry and setups that we've been using over the past 2 years. So on the left, you can see a general photo of the lizard lab when it's up and operational. And so we have the capacity to hold 36 individual 20-gallon tanks in there. And each of those 20-gallon tanks might be a little hard to see. Is my cursor visible at all? Wonderful. Well, in each of these 20-gallon tanks, we have a heat lamp in the back to provide a warm or a basking area for the lizards. And then we have a cooler area up front that is penetrated with UV bulbs. And on each one of these tanks, we also have a nozzle hooked up to a room-wide misting system. So we can ensure that all of the lizards have sufficient heat, lighting, and hydration. And all of those are hooked up to timers, so they're controlled electronically. So there is very little manual input, which is one of the nice things that we're able to do over in the lizard lab. And so the development of these setups certainly took a tremendous amount of time and tinkering over the past couple of years. As you can see on the right, we actually have the first, one of the first hatchlings that was produced in the lizard lab at the zoo. It's blended in quite well against this substrate. So for the past couple of years, we've been using locally sourced substrates, so essentially clay that is available on zoo grounds. And we started off by just using the clay straight from the ground as we found it, but we quickly learned that finding dime-sized hatchlings in a substrate that also contains small rocks and clay particles becomes near impossible. And so we've run into a number of these issues over time that we've been able to gradually adjust our husbandry protocols so that we've done things like sifting the substrate now so it's more of a sand consistency, and the lizards are able to burrow into it much more effectively than they were on the really coarse clay substrate. We've changed out the hides that we use on our natural or on our heated or basking end of their exhibits so that there is a natural rock surface there, which tends to retain heat much better even after lights and lamps have all switched off. We have adjusted our timing on misting systems consistently over the past couple of years, but have it down now. So we know that misting twice a day for 30 seconds aimed at the sides of the tanks rather than in the middle where the lizards are is actually more effective, largely because they seem to not be inclined to drink standing water. As more prairie or grassland specialists, they don't seem inclined to drink much water in their environments at all, but are willing to drink small droplets that are rolling off the sides of their exhibits, which may be emulating like a morning dew or something more similar to that, which was interesting to figure out as we went. And while it's not pictured here, another thing that we figured out in more recent efforts is that placing plants or artificial foliage like this sideways as opposed to upright actually increases usage significantly, as the lizards are more inclined to go burrow themselves in it or actually climb over it rather than interact with a standing plant as it's shown there. So throughout the entirety of the process of maintaining lizards for the past couple of years, we've been constantly working on and adjusting our husbandry parameters to try to maximize Texas horned lizard well-being at the zoo. And along with husbandry, we've been working on trying out how best to feed and grow Texas horned lizards, with the impetus behind the program trying to raise robust lizards for release back onto Tinker. We want to have sizable lizards that are capable of finding food on their own upon reintroduction. So one of the things that we found works really well is simply cast feeding fruit flies. And so here's one of the little hatchlings that had Drosophila melanogaster introduced into its little habitat, and they are quite inclined to go after small fruit flies that are walking around right in front of it. And we've worked on trying to provide some enrichment opportunities as well. So here's an example of a little feeder that I made by soldering holes in a small plastic container and then putting larger fruit flies, Drosophila hydei, in there. And so this seemingly mimics an ant colony a little more effectively than just fruit flies running around haphazardly. These soldered holes only allowed 1 or 2 fruit flies to come out at a time. And so the horned lizards got really good at just picking them off as they would leave their little plastic containers. And there we go. And we found that we had the most success with feeding out a variety of fruit flies and pinhead crickets, and that has worked seemingly fairly well for us for the past couple of years. So that's been the staple of their diet while in the lizard lab. And to focus on the microbiome aspect of the project, or the gut bacteria aspect of the project, one thing that I've been trying to do very frequently is collect gut microbial samples. And the best way to do that is actually through fecal sample collection. And the protocol that we ended up developing was simply placing a horned lizard in a small shoebox container, so like a 6-quart Sterilite tub, and feeding them in that once a week. And for whatever reason, if we could stimulate their metabolism while they were in a small 6-quart container, we were able to get fresh fecal samples fairly reliably. And this was a nice, effective way to also ensure that lizards were eating regularly, because we had Texas horned lizards housed individually in 20-gallon enclosures. It can be difficult at times to track all of the lizards and their dietary intakes. So here's just a quick video of one of the lizards that had a few pinhead crickets. And you can see here how there is just an array of small Texas horned lizards in 6-quart buckets. And so while it may seem a little primitive, it was actually very effective for finding fecal samples very regularly. And so we're hoping that this will pay off as we get into some of that data analysis a little later on. One of the really interesting things that I got to do while I was interacting with these horned lizards regularly is try to take mean weights, so weighing these lizards about every 2 weeks. And we were able to come to some really interesting preliminary findings so far. And one of the things that I find most surprising is circled in these red boxes. So we have a 2019 cohort, so a group of lizards that were found and raised from 2019 and then from 2020 again. And I have most of the winter months are in the square boxes, and somehow in some way, despite stable lighting, feed schedules, watering schedules, and temperatures, constant temperatures in the lizard lab, the lizards still decrease metabolic activity and slow their weight gain through winter months. And so that's one thing that I found was incredibly interesting, despite me giving them stable conditions year round. For 2 straight winters, the 2019 lizards did not gain weight for a period of time in the winter, and this was repeated in 2020. And so this is something that I'm really looking forward to breaking down a little further and trying to figure out why this might have been, because there seems to be some innate capacity for Texas horned lizards to refuse to gain weight in wintertime when they are not active or generally burrowed in over winter in the wild. This is a lot of text, but essentially to break down how the program has gone so far, we started with 2 initial cohorts, one in 2019 and one in 2020 for a joint release in 2021. And because we know we had variable survival rates between hatchlings and juveniles on Tinker Air Force Base, we wanted to look at how different age cohorts, or essentially sizes, because the year-old lizards were half the size of the 2-year-old lizards. So we wanted to release 2 different cohorts simultaneously to see how age and or size might impact release success. And in 2019, we started off with 35 eggs originally from 4 clutches, and we didn't take any wild hatchlings that year. We ended up getting 15 individuals to survive to that second year. But one of the caveats that we had is one of the clutches that we found in 2019 was flooded out by a thunderstorm the previous night. And only one of the 15 hatchlings that clutch produced ended up surviving all the way to release in 2021. So if we remove that potentially oddball clutch out, then we had a survival rate of 70% of our lizards from the time that they were recovered as eggs to the time that they were reintroduced to Tinker, which is double or triple what the survivorship roughly would be in a yearly average. And we had much better luck with the 2020 cohort as we had adjusted our husbandry parameters and had a better sense of how quickly we needed to find and move eggs over to the lizard lab. So we recovered 14 eggs and also moved 12 hatchlings that were found directly on Tinker Air Force Base into the lizard lab. And we generated a cohort of 19 animals, which was a 73% survival rate. So year in and year out, we seem to be increasing our survival rate in the lizard lab as we're improving our animal husbandry protocols. And we're hoping this continues as the project continues so that we can better increase the number of animals that we're getting back onto Tinker. And then this is covering some of the tracking protocols that Ray touched on a little bit, but essentially we had our 2019 lizards, our 2-year-old animals, all equipped with these new CTT tags, the solar powered tags that we were testing out this year. And then you can see these 1-year-old lizards are equipped with these harmonic radar diodes. And this is just because of the size of the lizards. We wanted to make sure that the tracking tags weren't more than 10% of the body weight of any given individual. And our larger cohort was significantly larger than our smaller cohort. So they were capable of housing or handling the slightly larger tags. But this is all 34 Texas horned lizards that we prepared for reintroduction back to Tinker Air Force Base. But before we let them go all willy-nilly and do their thing out there, we used the soft release implementation. So we built 10 by 10 aluminum sided pens on base that were dug in 4 inches underground. And then we put bird netting over the top, and we picked specific locations that had active ant hills and ant activity. We also baited them with basically fish and leftover salads from the Oklahoma City Zoo, which were used as essentially bait stations that increase the number of prey items that were in each pen. And we built 4 of those pens and placed 8 to 10 lizards in each one of them. And the lizards were left there to acclimate to conditions for 5 weeks. And at the end of those 5 weeks, we removed all of the lizards from the pen, and they became part of this ongoing managed and monitored population on Tinker Air Force Base. So here are a couple of photos of some of the larger lizards that were just hanging out in the soft release pen on one of the days in June. And so we initially moved all of the Head Start lizards on June the 1st from the lizard lab to Tinker Air Force Base. And then the second week in July is that picture on the right, where I was pulling lizards out of that soft release pen, and they were placed adjacent to the soft release pen after collecting morphological data. And then they were continuously managed and tracked as part of the native population, as I mentioned. And we've also continued collecting microbial samples. So every week at a minimum, we go out and try to find all of the Head Start lizards that we possibly can. And we do the same protocol that we did in the lizard lab, in that we place them in the same 6-quart Sterilite tubs in a nice shaded area. And for whatever reason, that seems to continue to be our best bet, is putting lizards in small shoebox containers and waiting a half hour or so. And we have done that since they were released, as I mentioned, in the first week of July. And to date, we had 34 lizards that had gone out, and we have 6 lizards that we're still actively tracking. So the hypothesized number of lizards from the Head Start program that are still on Tinker is around 16. That's our most optimistic number. And that is higher than the 6 that we're currently tracking, because lizards have been remarkably good this year at shedding diodes while we were in between captures, and they've also been very good at shedding their harmonic radar tags as well. So that's why we only have 6 active lizards being tracked, but we're cautiously optimistic that there are considerably more Head Start lizards still roaming around on Tinker Air Force Base. And with that, this is a nice segue to what we're planning to do next. And we actually started with the development of the 2021 cohort. So we have 13 eggs that recently hatched out. They hatched out last Thursday, so they're a week old now. So we have 13 eggs that hatched out, and then we also brought over 10 hatchlings this year to develop a 2021 cohort. And so we are planning to do a second release in 2023. And thanks to the Oklahoma Department of Wildlife Conservation, we have the capacity to do that. And we're hoping to use the data that we've collected in terms of morphology, behavior, and husbandry protocols, essentially, and compare them to a second iteration of this release protocol to see what we're doing and to determine if it's working and to figure out what we can be doing to improve the conservation outlooks for the head started and released animals. And the last thing is in terms of the microbial data and analyses, we've collected 250 fecal samples, I want to say it is, over the past couple of years. And so the unfortunate thing about that is that we will be having to analyze all of that data in one fell swoop. We have to get all of the sequences sampled at once. So I'm hoping actually next month that we're going to be starting the process of sequencing all of those fecal samples and then determining what kind of gut bacteria may be at play for this reintroduction program. And I think that is all I've got.
Hey, thank you, Sam. There is one question, which I think you kind of touched on some of these things, but if you could just kind of briefly address this. The question in the chat is, how old are the oldest horned lizards that you're reintroducing, and what are your release strategies? The chat says that the San Antonio Zoo releases a variety of ages from over a year to hatch in a single release and release them in a variety of locations. On the Dallas and Fort Worth zoos release 50 to 100 hatchlings typical in a single release and release in a similar location. So I guess, what are the ages that you're releasing them at, and are there release strategy, I guess, location more so than Tinker, or just Tinker for now?
Yeah, good question. So with this first implementation of the Head Start program, we did not release hatchlings directly. We released animals that were hatched out in 2019 and then those that were 2020. So lizards that were about 2 years old and 1 year old respectively. And so those were our 2 cohorts. And I know that other programs have done differing strategies. Like I think Dallas and Fort Worth are breeding animals in their facilities and then releasing hatchlings within 8 weeks. What we were trying to do with this program, having the wonderful array of data and published information that we have, is we know specifically that hatchling and juvenile mortalities are really high. And we wanted to see if we could raise animals past those large mortality stages and get them to a greater survivorship year over year by releasing larger and more robust animals. And so we did the 2 cohorts that we had, and they were both released right onto WR3, or the Tinker property that Ray had been showing maps of on his presentation.
Okay, thank you very much. Okay, so we are definitely running low on time. But I don't see any reason why we can't continue with our last speaker. I do understand if you have other obligations. We will be putting this presentation on our YouTube channel as well for future viewing. But I do have one more Oklahoma presenter for you, Alex Fulton. And Alex is a new member of the Seiler Lab at the University of Oklahoma. She joined in May of 2021 as a master's student focusing on Texas horned lizard research. She is interested in the conservation of reptiles and amphibians and research with direct implications to help further that goal. Prior to starting to work in Oklahoma, Alex worked for a variety of agencies in different states, but most recently, she worked for the USGS in California, managing western pond turtle and giant garter snake projects. And she's going to talk about her her new project working with OU and kind of stepping outside of Tinker Air Force Base and looking at the potential for horned lizard conservation research in other parts of the state. All right, thank you.
All right, can you hear me? Okay, well, thank you so much, Miranda, for the introduction, and thank you everybody for having me speak here. I am new to Oklahoma and I'm new to Texas horned lizard research, so I'm very excited to talk with a group that is definitely more familiar with Texas horned lizards than I am. My presentation is a bit on the shorter end, so I'm not going to keep you too terribly much longer, and I will kind of try to move through it at a decent speed, so don't worry too much about that. As Miranda mentioned, I'm going to be talking today about Texas horned lizard research that's taking place in western Oklahoma, specifically at Beaver River Wildlife Management Area. So I'm going to be talking today about the first year of a 3-year study that we're working on. Okay, before we get going, I just wanted to acknowledge some people that have helped out with this research so far. First and foremost, our funding is provided by the Oklahoma Department of Wildlife Conservation, and we wouldn't be able to do any of this without that support. And I wanted to give a huge thank you to the staff of the Beaver River Wildlife Management Area. I really cannot thank them enough for all of the help and support that they provided this summer and making sure that our research was able to run smoothly and helping give us lizards and share all of the information. It was wonderful to have that great welcome. And then also the 2 summer field technicians that helped me with this research this summer, Grace Payne and Whitney Buxton, for all of their work on the project. Okay, so this 3-year study that we just started last summer has 4 main objectives. The first is to estimate the distribution, population density, and habitat requirements of Texas horned lizards at 3 WMAs in western Oklahoma. So last summer we were at Beaver River Wildlife Management Area. The next 2 summers, we're going to go to 2 different wildlife management areas. We haven't decided which ones we're going to go to for sure. We are still deciding between Cooper, Sandy Sanders, and Pack Saddle. The second objective is to develop and test standardized survey protocols for locating and monitoring Texas horned lizards. That's something I'll be talking about later on in the presentation. The third is to evaluate the status of harvester ants and red invasive fire ants. We do that using little insect pitfall traps that I'll show a picture of further on. And the fourth is to analyze population genetic diversity of Texas horned lizards. So the way that we're able to get these DNA samples actually works really well, because every time we capture a lizard, we not only measure the spines and the mass and the length and that sort of standard stuff that you would expect, but we also give a unique toe mark that then lasts for the rest of their life. Whenever we do that, we actually are able to take the toes and then keep them for further genetic testing later. All right, for those of you that may not be familiar with Oklahoma, or especially the Panhandle, the purple dot is generally where we were at. We were in Beaver County, at Beaver River Wildlife Management Area, which is near Beaver, Oklahoma. That is where we spent the summer. We were there from about late May to early August this year, and most of our effort went into drift fence trapping. So we used these what are called Y arrays, and we deployed 10 of them throughout the wildlife management area. If you're not very familiar with these, they're made up of a series of drift fences, funnel traps, and pitfalls. You have a center pitfall trap with 3 wings coming out and funnel traps on either side. We check these 2 times a day. That was very helpful. We went out in the morning, and that was a good opportunity to get amphibians that might have gotten in overnight. I'm new to Oklahoma, so I don't know how typical this was for panhandle weather, but we got sporadic rain throughout the summer. And we would get a lot of spadefoots, sometimes tiger salamanders. Doing a morning check made that so they did not desiccate throughout the day. And then we also did an evening check, during which time we were able to get the critters that got in during the heat of the day. And kind of once it started, the ones that got in once it started cooling off. And that way they didn't have to spend excessively long, overnight in the trap. We did process all reptiles that got into any of these traps and ones that we found opportunistically, not just the Texas horned lizards. I'm sure some of you have installed drift fences before and fully understand that it is a ton of work. It is a lot of effort. We were really lucky that the soil out at Beaver Wildlife Management Area is kind of like a fluffy, sandy soil. So it actually wasn't too terrible to dig in. And for the most part, it wasn't too terribly rocky. But because the soil was nice to dig in, that also meant that our arrays weren't always the most stable. So we spent a lot of time this summer really reinforcing the arrays because they tended to shift in the soil a little bit. This is what the finished product looks like. As you can see, the wildlife management area is absolutely gorgeous. It was a beautiful place to be for the summer, just, you know, sprawling views, very green, lots of diverse plant community, lots of sage, yucca, various cactus, a whole bunch of other plants. This is another one of our arrays. Some of our locations had more like the really bright red soil. A couple things I did want to point out is this was the first year of our study, so we learned a lot about a lot of things out there. And one thing that we played around with this summer was altering the shade that we have over the traps. So you can kind of see in the middle here, this is a funnel trap that's along the side. We had a piece of wood over. As we moved into more of the heat of the summer, it just got really hot during the day. And it didn't cool down very much at night. And so we made these larger pieces of shade wood that you can see here that we actually deployed on all the traps just to provide a little extra shade, a little extra insulation. Another thing that we played with is the 5-gallon buckets. We had these large pieces of plywood that we put over top, which provided really great shade, but I saw sometimes animals running over top of them. They could kind of get a little bit of motion going and just hop over the holes. So later on in the summer, we kind of put them up on little stakes so that they provided really good shade, but they're more of an elevated platform. And then animals could not just run over the bucket, and they could also get in from other sides easier. There is a lot of grazing cattle on the wildlife management area. We had 2 different types of cattle that we were working with. What you see here is kind of in the southern section that we were in. There was a lot of stalkers, which I was not familiar with that term, not being from Oklahoma, but that is apparently like teenage boy cattle. And they were up to no good very often. They really were curious with our drift fence arrays. As you can see here, they love to sniff them. Sometimes they would kick our funnel traps. They just like to rub against them and knock them down. So we spent a fair amount of time cleaning up after the cows. Luckily, in the other section of the WMA that we were tracking, it was cows and calves, which were far more respectful of our traps. They would spend time in them. We would, walk up to the traps and see them laying around, but they were not destructive as these stalkers were. This is a picture, not a great one, but a picture of our insect traps. We basically use these like little deli cups, and you put the lid on it, and then right under the rim, we drilled or paper punched a series of 12 holes all the way around it, and then dug a little bit into the ground and filled it in. And it's a pitfall trap that's specifically for insects. And in the bottom of it, we put propylene glycol, which is an oil, but luckily it's not anything that's super scary. I even saw in my sunscreen this summer, propylene glycol was an ingredient, so it was something that was relatively safe to work with. And then the insects come in, fall down, they get stuck in the oil, and they can't get out. And so what we did was every week we swapped these out, and we had at each of our Y arrays, we had 3. Between each of the pairs of wings, we put one randomly, and then every week this summer we went out and we emptied it out and replaced it with a new one to start fresh. We haven't had time yet, but the goal is to go through all of these and identify and be able to kind of characterize the insect community at these locations. We also did an extensive vegetation survey at each one of our arrays. What we did was we had a rope and we put it out 20 meters, and we used this 1/2 meter by 1/2 meter Daubenmeier frame that we made out of PVC. And every meter along the rope, we alternated sides and did a full vegetation analysis going down to species. So we got a lot of really fine scale detail for that. In this picture is the technician, Grace, who was absolutely amazing with plant stuff. And I was very grateful for that because that is not my strong suit. All right, so here is a map. Beaver River Wildlife Management Area is actually the second biggest wildlife management area in the state of Oklahoma. So it's massive, and it goes even further off this map that you don't have, but we didn't have time to drive all the way around. So we clustered our traps in 2 separate areas. So 1 through 4 over here, that's kind of where that red soil picture was that I showed you. And then 5 through 10 was in the western section. And it's difficult to see from this picture, but we did pair our arrays. So we kind of had 5, 6, 7, 8, 9, 10. And so we'd park at one location and be able to check 2 arrays there, which was also, it was good to be able to have multiple places with similar habitat. In this map, I'm showing all of our horned lizard captures throughout the summer. The triangles are pitfalls and the squares are funnels. So those are ones that are associated with our drift fence arrays. We actually only had 2 in pitfall traps and 2 in funnel traps the whole summer. So that actually didn't work super well for us. And we had far more captures with opportunistic, which typically was just driving on the road and seeing them, which, you know, many of you know that's a great way to find them. And we had a lot of luck with that. The 2 points that I wanted to get across with this map is they were all over the wildlife management area. They weren't just clustered in one area, they were pretty widely dispersed. And these were just the areas that we spent time in. I expect that if we put a lot of driving or other effort into other areas in the WMA, we would have found lizards other places. Not everywhere is very accessible though. It's a very big area and there's a lot of roads that are not easily passable. I also wanted to point out with this map that it wasn't that the places our traps were, there weren't lizards, because we were opportunistically finding lizards all around our traps. They just weren't getting into our traps very well. So this is kind of a zoomed in picture of that, showing on the western section where our different capture types were. And you can see, you know, we're getting opportunistic captures going to and from our traps. Likewise over here, we had where these blue dots are, for the opportunistic captures, we actually had pitfall and we actually had, we had the Y arrays there, but they just weren't getting into those traps, but they're on the road near the traps. All right, so these are our total numbers for the summer. As I mentioned, we had 2 in funnel traps, 2 in pitfall traps. We had 4 in road cruising surveys. So we were kind of strapped for time this summer, but as we started getting towards the end of the summer, we were able to devote more time into looking into alternate methods for surveying. And we did 4 road cruising surveys that were 1 hour each. And we were able to get 4 lizards through just those 4 surveys, which, you know, considering all of the effort that we had with the drift arrays throughout the summer was 4, you know, that's definitely a pretty good bang for our buck with that, but the vast majority was from the opportunistic road cruising. So that was primarily the wildlife management area staff finding lizards for us. They did a lot of driving around in their duties, especially with tractors, doing various road work, filling in holes, that sort of thing. And I've never seen them from a tractor, but I've been informed that they're actually really easy to see. It's something about being up high and being able to see down. You can see their movement, you know, even more so than a truck. They'd run out to get them. And then we did find them, you know, driving to and from our arrays as well. All right, so moving forward, like I said, in the next 2 summers, we're going to be going to 2 different WMAs. We're definitely going to continue to test and look into protocols. So we're not going to give up drift fence trapping altogether, but next year we're definitely going to reduce the amount of drift fences that we have out there and maybe think about some different configurations, and then be able to devote more of our time to the foot searching and the road cruising and see how that goes. So definitely a lot more to explore. This was just the first year of the study. So a lot more coming up with that. And that is all I have. I don't know if we have time for any questions.
Thank you, Alex. I was very excited to hear about that because I was still in the silo lab at OU when this planning started with this project, and I graduated before we could get it off the ground. But I was very interested in seeing that. That was all new information to me. So very cool. Thank you for sharing that. At the moment, I've addressed all questions in the comments. I think if I have not, please go ahead and do that again. But we've already gone over time. So like I said, if you want to watch this again or share it with any of your family and friends, it will be posted soon on the HLCS YouTube channel. Thank you to all of my presenters, and thank you to everybody for being here. We are all very sad that we couldn't all meet in person and have our conference this year, but for everyone's safety, we thought this would be a great alternative. And this series is going to continue. So we're going to do the third Thursday of every month. We are going to have another series. We're going to focus on a certain state or a certain region. And so be sure to keep up with our Facebook page or read our newsletters that we email to you for more information with those going forward. That's all I have for you. Thank you. I don't know if Mason or anybody else has anything they would like to add, but either way, thank you very much for joining us.